Well, thank you very much for the introduction, although I didn't understand it. I hope it was not too bad. So I'm a medical doctor by training, but all through my life I've been interested in medical genetics and molecular genetic research. So don't trust me as a clinical medical doctor, but trust me as a molecular genetic scientist. And I'm affiliated with these two universities in Denmark, but also with Wada that you have heard so much about. And through the years, I've been involved. There's more than 30 years we have collaborated. It tends to be more and more time for me in China, and when you have heard Yang Huanming's talk, you understand why. The resources that we have in Wadajin is second to none. So you really can do things in Wadajin that you cannot do anywhere else. So being a medical doctor, I'm of course interested in problems of health. And I think it's fair to say that we are in the middle of a health crisis because of the rise in chronic dysregulatory and degenerative disorders. So diseases like atherosclerosis, diabetes, hypertension, dementia, and cancer are rising all the time, and it costs us more and more and more to take care of all these patients, and as they live longer, they tend to get one disease after the other, so eventually they have 7 or 8 of these diseases, and it becomes very costly. So there is a problem that people refuse to die, they just get on getting more and more of these degenerative disorders. And it doesn't get any better because we have adopted a lifestyle that is not in balance with our genes. So there are so much in our society that doesn't really fit us. And one thing, of course, is that we overeat. So the genes that were good for us when we were starving and doing very hard physical work in very harsh surroundings, those genes have become dangerous for us now because we have adopted a totally different lifestyle and we, as I said, are not short of food and calories. So as a geneticist, one can think about the genetic background of diseases, and this is a hypothetical curve, but it illustrates my point. So we know about these inherited monogenic disorders, and these are the well-known but rare diseases like inborn errors of metabolism, different types of dystrophies, and so on. And then we have a period when we are young where we are doing quite well. Because although we know that we have inherited quite a lot of rotten genes, so to speak, we are able to handle it because our cells are young and our organs and our cells are young. But when we start to reach 60, then there is a problem. Then suddenly it becomes very important if we have chosen our parents right, sort of. What genes we have is starting to become more and more important, and eventually, of course, we die. So that is 100% genetics, right? Nobody has lived beyond 120. So the question is then, can we in some way remedy this problem, decrease the impact of these severe monogenic disorders and keep this curve down until suddenly everything breaks down, right? So my ideal is stolen from the first car that I had. I bought it second-hand in Sweden for almost nothing, and it was running year after year with very simple maintenance until suddenly it broke down totally. I couldn't even get to a mechanic. A friend of mine came and said, there is absolutely nothing in this car that can work or can be rescued. So then, of course, I think that was a very beautiful death. So I would like to die in the same way as my old car, right? So how do we do something about this from a geneticist point of view? Yes, one way, of course, is to try to understand what are these gene variants that are creating problems for us when we grow older, and by knowing a little bit about those gene variants that make us susceptible, perhaps we can counteract their effect in the future. So therefore, a lot of people have used large-scale sequencing of the human genome in association studies to find these, what I would call, illness variants that give us susceptibility for these degenerative disorders that I've just talked about. And this is done traditionally in case control studies, so you have a group of patients and you have a group of controls, and you look for association in what we call a genome-wide association study. This is being done a lot all over the globe. The problem is that the method is not very strong. You need an enormous number of patients to get significant results because you are looking all over the genome, and you rely on things like linkage disequilibrium and other things that I won't have time to go into here. So it's not a very strong method, but it has given us a lot of hints as to what these susceptibility genes are, or susceptibility gene variants are. And this will help us in the future, I think. But another approach that I find more interesting is not to look for illness variants, but wellness variants. And one way of doing that is what we call looking for genetic superheroes. And these are people who have genes that should make them very ill and perhaps have led to death long before. But they seem to be resistant. So the monogenic disorders that we know very well, they are very good to look at this. And one example is there was an 80-year-old lady who came to the doctor. She had only one problem, and that was she had a big lump on both her Achilles tendons, and that created a problem. She could not put her dancing shoes on. And when the doctor analyzed this, he found it was xanthomas, that is, they contained enormous amount of cholesterol. And as a clever doctor, he asked about the family, and there were lots of people in the family who had died very young in cardiovascular disease. And when we analyzed it, it turned out to be a severe form for familial hypercholesterolemia. And she had the gene that should have killed her 60 years earlier. But she was still running around, only having the problem not being able to put the dancing shoes on, right? So she was able to get the cholesterol out of the blood, and she stored it in her Achilles heels. She also had xanthelasmas on her eyelids and other things, but she was very, very efficient in removing the cholesterol herself. And this patient and many other of these genetic superheroes are now studied to try to understand this mechanism of resistance, so we can learn something from these rare individuals. But we can also look for variants that give wellness by seeing situations that select for genetic resistance. And one such example is resistance to hypoxia. This is a big problem in old people because they often don't get enough air by breathing because they have chronic obstructive pulmonary disease, COPD, or other types of lung problems. So our question was, can Tibetans who live up on 4,500 meters and higher for many generations teach us something that can benefit patients with COPD and other problems of hypoxia? So we made some years ago, it's almost 10 years ago we started actually, sequencing humans, and we only sequenced 50 humans, so it was very unlikely that we would find anything really. I was not sure this was worth it. But we found very, very strong signals to our surprise. So the strongest signal was from this type of protein, which is a transcription factor, EPAS1. And this is the strongest selection ever seen in the human species, actually. And you can see this on this picture, that you have the controls on this axis and the Tibetans here, and you can see that there are many variants that are more common in the Tibetans than in the control populations, especially down here in this region. That is variants that are very rare in controls. But this one really is very clearly selected for in the Tibetan population. And then you can do various types of studies, like you can calculate a statistic that we call FST, and the branch length between the different population for this gene shows you how strong the selection has been. So there has been a phenomenal selection in the Tibetans if you compare with the average between these three populations. These are Danes that we use as outlier controls, and we are definitely not adapted to high altitude. Denmark is one of the flattest countries in the world. So this is just phenomenal. And one reason for this is that it now seems to be that the ancestors of the Tibetans have had some sexual contacts with archaic hominins. There is a group called the Denisovans, and the block in which this variant is present seems to be derived from the Denisovan hominin population. So this is an example of something that can speed up the selection. You get a gene from the outside, perhaps in many copies in the population, and thus you can select very fast for this, because the time span between the Tibetans and the control lowland Chinese is not that long. It's only a few thousand years, or 4,000 years perhaps. So this is an example where we can find some variants that seem to be protective against anoxia problems, and of course these variants are now studied in molecular detail. So another thing we can try to do is we can think about the resistance as just a variant of susceptibility. So healthy aging can be regarded as a complex disorder, right? So only a minority of the population can survive for a very long time without cognitive dysfunction, without diseases. So it is realistic and relevant to study these oldest old successfully aged people. And you can also look for somatic mutations and other epigenetic patterns that are selected during healthy aging with such a group. And for this, I think the Chinese population is unique because China has the largest and most homogeneous population of all countries in the world. And homogeneity is very important here because you have to have an ethnicity that is rather similar in order to find these variants. And there is no shortage of successfully aged oldest old in China. There are hundreds of thousands, and there is a general interest in healthy aging and aging research in China. So we have joined forces with some professors in Beida, especially Professor Zheng Yi, who is not a geneticist, but is a demographer and economist. And he has led a fantastic project that is called the Chinese Longitudinal Healthy Longevity Study. And it has gone on since 1998, and 7 waves have been performed. And in the last wave, we also managed to persuade them that they should take blood samples so that we could do genetics. So now we are increasingly involved in this study. But as you can see, they have done a fantastic job with 90,000 interviews to see how well these people function, and we have more than 15,000 people that are more than 100 years old and so on. So this is a fantastic material, and we have started to analyze this. We are by no means ready yet, but we have analyzed more than 2,000 cases and controls, and I won't go into the details about the setup of the analysis, but we use the South China first as a discovery group, and then the south part as a validation group, and vice versa. And we find a lot of interesting variants, and if you make what we call a Manhattan plot of all the chromosomes from 1 to 22, these dots represent variants that are very much enriched in this successfully aged population. And if you look at pathways and networks that are enriched, it's primarily pathways that have to do with immunity and inflammation, but there are also some kinase networks and surprisingly calcium homeostasis and signaling. And of course we know that there is a lot in the metabolism that is important in order to be successfully aging. And we can also start doing gene-environment interaction studies. So this is just an example of 2 transcription factor genes that show variants that, together with tea drinking, which of course is a very important thing in China, and where traditional Chinese medicine has said that this is good for you. And I think we can clearly say, yes, it's good for you, especially if you have special FOXO genotypes. And we think that this has to do with the large numbers of polyphenols that are present in the tea. And we know that some of these polyphenols interact with these transcription factors, and there's especially one epigallocatechin gallate that we are now testing in molecular experiments together with FOXO1A transcription factor variants. So these studies give you a lot of clues to what things you can do and what you can perhaps utilize in the future to improve the situation with respect to these degenerative diseases. But it's by far not enough, so this is just a preliminary thing to do. So what we mean is that we have to learn to do regenerative medicine, to move this curve from here down to here, right? And regenerative medicine, we know, is possible, but we are rather bad at it. So other animals have a much better regenerative capacity, like amphibians and fish and so on, and we are really, really poor. But on the other hand, it's quite clear that we are not totally incompetent, because when we are newborn or in utero, we can repair much, much, much better. So, for instance, a heart defect which leads to cell death can be repaired perfectly in some cases. And the process is a combination of inflammation, where you remove the bad cells, and a cell replacement where new cells are coming in and produce the correct types of cells in the tissue, so you have a functional restoration. And if you remove the macrophages, then you just get a scar and dysfunction. So. And in the adult, unfortunately, there is nothing you can do, right? But in other animals, this can be easily done. So this animal, the salamander, which is an amphibian, is really, really good at this. So if it got that heart defect, it would immediately make a new heart. And if its eye is destroyed, it makes a new eye. And if you cut one of its limbs up here, it makes a little bud with undifferentiated cells that called blastemas, and from that bud, a new functional arm, or this is, I suppose, the foreleg of this animal is growing out. So it's amazing what these animals can do. And of course, we would like to learn from these animals how to do it, because it would really revolutionize medicine if you can do regeneration that efficiently also in humans in the future. So some people have started to work with these salamanders, and there are two main issues. One is that if you cut down here, you get a distal blastema, and this knows that it should only make the foot or the hand or whatever you would call it. So it knows its position. If you cut up here, it makes the whole limb, right? So there is a positional effect here that makes these undifferentiated cells know where they are and what to do. And the other thing, of course, is that these cells, from a rather undifferentiated stage, they start to organize themselves. Not only the individual cells, but it can interact with the others in the blastema. So you do a skeleton or the bone, and you do the muscle, and you do the vascular system. So this is a very interesting model system to work with. The problem is that we don't have the genome of these animals. It is gigantic. It's more than 10 times as big as our own genome. So it's 32 gigabases, 32 billion bases. And there are regional duplications that have occurred quite recently. So it's very, very difficult to sequence and assemble this type of genome. We are trying to do it, but before that, good groups like this group from Harvard have started to sequence all the RNA that is present in different types of tissues and what is happening during regeneration. So this is the workflow. And then at the end, you come up with some candidate genes. You don't know much about it because you don't have the genome and you don't have the homologous regions in mammals. So, but there are a couple of genes here that you can knock out and see that if you knock out these single genes, the blastema either dies or it doesn't develop properly. So there is a faint start here, but we have a lot to do. So then, more than 50 years ago, I was working with cloning of amphibians. So this is a clone of albino frogs that have been made by taking eggs from this frog. As you can see, it looks very different. And then the nucleus has been taken out of that egg, and instead we have put in the nucleus from an albino line of frogs. And as you can see, you get a clone of frogs. So this was one of the first examples of successful cloning from somatic cell nuclei. And then many people, including us, tried to do this with mammalian species, and we were not successful. So it took more than 30 years before suddenly Ian Wilmot and his group at the Roslin Institute in Scotland managed to clone the first mammal, the sheep that was called Dolly, after Dolly Parton. and this is the surrogate mother sheep. You can see it's a completely different breed. But this is Dolly, and Dolly then could give birth to offspring and so on. So it was a functional sheep. So this showed us that somatic cells, even in mammals, we had started to doubt that this was possible in mammals. In the textbook, sometimes it was said that this can only be done in amphibians because they have such a regenerative capacity. But this turned out to be wrong, so it's only a technical problem. So any somatic cell, even from our body, can develop into any other cell types. There is no doubt about that. But we don't know exactly how to do it, except for this case where you change the cytoplasmic environment, right, by somatic cell nuclear transfer. We know that this has to do with the programming of the genome, and this we call the epigenome because these are epigenetic effects that lie on top of the DNA sequence. So here is a simple cartoon of the packing of the DNA. So here you have the DNA strand, and you have 2 meters of DNA in each cell, and you should pack this into a nucleus that is only a few microns big. So it has to be packed a lot, right? So first you wind it up on nucleosomes that are made of of 8 histones of 4 different types, and then you get the bead of a string on a string, and then this is condensated into a fiber with a 5th histone, and then it's organized in loops of this 30 nanometer fiber, and these loops are turning out to be very functionally important, because in this way you can put genes that are actually quite far from each other very close to each other, and you can put regulatory elements that are very far from the gene very close to the gene, so you can change the gene control, and this is important for epigenetic regulation. And then you have even higher order chromatin folding, and some of this folding leads to torsional stress and so on that change the properties of the DNA. So it's very interesting right now to find out exactly what is happening during this higher order reorganization of the chromatin. We know some things that can affect this. So the histones can be modified by epigenetic factors, and there are a lot of different types of histone modifications, and we understand a few of them, what they are doing, but most of them we don't understand at all. And you can also methylate the DNA in various ways, and in this way change the way the DNA is packed down. And finally, we now start to understand how you can make a loop from here to here, so you change the superstructure of the genetic material. This happens in a regular fashion during cell differentiation. And although all these things are reversible, they are still rather stable, so that the daughter cells of a cell with a certain epigenome has approximately the same epigenome. But it's very difficult to study because the epigenome is also affected by factors that are outside of the cells and even outside of our bodies, and aging itself is creating spurious stochastic methylation. So you have a background that is complicated on which you have to find those epigenetic changes that are functionally important. But we are very interested to do that because we know that there are a lot of diseases of the type that we have just talked about that are clearly associated with epigenome variants. So it's actually even stronger association between epigenome variants in certain regions than it is with genome variants in the same region. So as a geneticist, we have to face the fact that the DNA sequence might not be quite as important as we thought in medicine. Epigenome changes are at least as important and probably more important. So this means that we should work to manipulate the epigenome and learn about this reprogramming. And we start to get some technologies that I will return to. You have probably heard about CRISPR-based technologies. And this makes us very, very excited because now we can do it in a more specific manner, and this opens up for regenerative medicine. So in regenerative medicine, we like the pig. So we think the pig is a very good model for the aging man, and probably also the aging woman, but I don't dare to say that. It is very similar to us in physiology and metabolism. It eats everything, and it has a size and longevity that is good because we can follow what's happening during the generative process, and we can intervene and see what is is happening. So, and we have also, of course, sequenced the whole genome of many different breeds of pigs, and it's quite clear that the pig is genetically much closer to us than our rodents and other of the normal laboratory animals. So it's much easier to compare and go between the pig and ourselves. What we are doing is that we genetically design pig models for these chronic degenerative processes. The objective is to understand the process itself and to develop early diagnostic tools of the degenerative process, and of course to develop new forms of disease prevention and treatment. So you have a disease-prone pig. It's not ill, so it doesn't have the disease, but it has the susceptibility to develop disease, but you can prevent it and make it perfectly healthy by various means, right? So the way we genetically design pigs is by using a variant of this somatic cell nuclear cloning. So we take ovaries from a pig and isolate the immature eggs, and we mature them in the petri dish, and then we cut it in a very simple way and throw away the part that contains the genetic material, right? And then we add genetic material to this empty egg. And this genetic material we derive from, from some type of cell from a donor pig. And we genetically modify and select and analyze, so we know that this pig genome is genetically modified in exactly the way we want. And then we just fuse this, so we get this into the empty cytoplasm, and we add another piece of cytoplasm to make the reconstituted egg big enough. And then you activate and you grow in a petri dish this little reconstituted embryo into the blastocyst stage. And this blastocyst stage can then be used in various ways. One is you can just isolate the cells of the inner cell mass and grow them and differentiate them out into different lineages, and thereby learn something about what your genetic modification has done at the cellular level in the different types of differentiated stages. But more interestingly, you can surgically transfer this to a surrogate sow that then gives birth to a clone of pigs that is genetically different from the sow, but is from this genetically designed fibroblast population, right? So this is very interesting, and here is just an example for our firstborn pig that we of course named George Clooney because it was the first clone pig, and this is just to recognize Gabor Vajta, who was the person who really developed this method into a very efficient way of creating genetically designed clone pigs. And this is to show that many of the people working in this group were Chinese, very skilled Chinese students without whom we would never have developed this technique as well as we have. And this is just to show that it's a very efficient method. So this is we call this cloning method handmade cloning, because you just cut, you don't need any micromanipulation or anything like that. And here you can see we have cloned two types of pigs in the same in the same sow. So this opens up for the possibility that you can clone some male pigs and some female pigs with the same modification, and then when they are 5 or 6 months old, you can cross them, so you can immediately get offspring that is genetically designed in the way you want. So the efficiency is very high. We get more than 50% reconstitution rate, and we can get 10 piglets from one sow, and very many piglets from each transferred embryo. So compared to other cloning methods, it's quite good. And we have used this for many different things. We have tried to create models for degenerative disorder by designing pigs that have the amyloid precursor protein with a so-called Swedish mutation that gives early onset Alzheimer in humans. And we have also a presenilin 1 gene with this particular mutant that is also associated with early onset Alzheimer. So we have done several studies, but we don't have any spectacular phenotype yet. But here we have quite a spectacular phenotype. This is a collaboration with a pharmaceutical company, Leo Pharma, who is interested in skin inflammation, and just by causing the pig to express these integrins ectopically, we get a very severe skin inflammation. Unfortunately, it's so severe that it's not ethically reasonable to let the pigs suffer. So we are now trying to decrease the dose so that they will not be that ill, and then this can be done, be used for trying to manipulate this unhealthy inflammation that occurs. And we have also made some cancer models that are not particularly interested, but we have done quite a lot in atherosclerosis. So we have made a set of pig breeds, or whatever you would call it, that have these type of genetic defects. And one example that I want to say is the PCSK9 gain of function mutated pig. So PCSK9 is a molecule that is synthesized, and it binds to the LDL receptor, and this helps it internalize the LDL particle, the lipoprotein particle. And normally this should be internalized, and the LDL should be degraded together with the PCSK9, but the LDL receptor should be recycled so that the concentration of on the surface stays high. But if you have this mutation, which is illustrated by this yellow flash, then this binding becomes irreversible. So this means that both the LDL particle and the LDL receptor is degraded in the lysosome. So you end up with a cell with very little LDL receptors on the surface, and this, of course, leads to hypercholesterolemia. So this is just some examples of some founders, pig, F0, you could say, the first cloned pigs, and you can see that the plasma cholesterol is increased, and if you give these pigs a diet which is high fat or high fat, high cholesterol diet, this concentration becomes really, really criminal. And if you analyze the LDL, the particles, the lipoprotein particles, you see that most of it is this dangerous LDL that we know is causing atherosclerosis. So this is also inherited to the offspring, and you can see that the levels of the cholesterol is just extreme compared to the control levels that are very far down. And these pigs also develop atherosclerotic lesions that look very much like the lesions we see in humans. And already after 40 weeks, we see very severe such lesions. And this is an example from a 1-year-old pig that is really, really severely affected. But these pigs are now used to develop non-invasive diagnostics, new contrast compounds that makes it possible to see these early atherosclerotic lesions and judge how far it has gone. And this is a very important clinical problem that you have no such methods, so sometimes you don't understand why people suddenly die of cardiovascular insults, and when you look, they have very severe atherosclerosis all over, right? And it's also used to develop methods for preventing early lesions and perhaps even reversing early lesions. And also people use it for new types of surgical treatment of late lesions. So this is one example. Another example, which is a collaborative study between the pharmaceutical company Novo Nordisk and Aarhus University, and we have formed a little company ourselves that we call PixieGene. And this is to develop models of diabetes. And here is one example of such a model, and you test it by making a glucose tolerance test. So you inject a lot of glucose and you see what's happening. And as you can see here on this picture, a normal pig would secrete a lot of insulin when it's challenged by such an intravenous glucose amount. But these pigs, they cannot do that at all. So they are now almost totally unable to induce insulin secretion. And the difference is really very significant. But to our surprise, still these pigs are able to clear the cholesterol quite well. Not quite as well as the normal pigs, so it's the slope of the curve which is important. So the slope of the curve is a little smaller in the genetically designed pig relative to the other pigs. But still, I mean, it's really, I mean, human beings would be dead if they had this situation. So somehow the pig has a resistance, and we think that part of this resistance lies in the kidney, so they are much better at secreting glucose through the kidney than we are. So that's why they can keep the concentrations relatively low. And we also think there are differences in the liver, so they take up glucose in the liver without help of insulin, another mechanism, and transform it into other compounds. So we are now hoping to study these pigs to learn how they are, why they are so resistant. So another thing that Yang Huan Ming mentioned, we must not forget our other genome. So it is clearly of great importance for our health, right? So this was just an article that he also showed. And to manipulate with the genome, we have made some micropigs based on our knowledge about the Laron syndrome in humans, where you have proportional dwarfism due to mutations in the growth hormone receptor. And these pigs are really, really very cute. So you can see I like it and it likes me. But these pigs are made to be able to intervene in the gut microbiota, and we have sequenced the gut microbiota in pigs, so we have something to work on, and it will be interesting to see if we can really change this in relation to various types of disorders of the type I've talked about. So these pigs will also be used as models for regenerative medicine. So since they are cloned, we can clone another pig from the same original fibroblasts, but without the genetic design that we have here. But this means that they come from the same original donor, so there should be no rejection or histoincompatibility between them. And then, of course, you can label these cells and use them for transplantation directly, or you can grow cells from them and differentiate them in various ways so they become suitable for cell therapy, right, of the pig. And eventually we should take out cells from the susceptible pigs and do the same and correct them and put them back in. And of course, eventually we would like to do that in the pig itself without taking out anything. Just make sure that the healthy cells have a chance to grow and the bad cells are selected against, right? And to do this, we are working with two methods: single cell sequencing that you heard a little bit about, and the other is genome engineering, and this is both genetic and epigenetic engineering. And the goal is to be able to study the evolution of somatic cells. That is, what somatic cells in the tissue have a growth advantage or an advantage relative to the unhealthy cells. And this, of course, will be very important for regenerative medicine. And this is my favorite quotation, that if you really want to understand something, you should understand the evolution of the biology. And somatic cell evolution, I think, is an area where we are very, very ignorant at the moment. But this guy, Dobzhansky, he could see that many, many years ago. So this is just some articles that we have published on single cell sequencing, and I think Huan Ming even showed that if you look at a number of cells from, in this case, a bladder cancer, you can classify them in certain groups, right, due to the pattern. And this means that you can reconstitute somehow the history of the cell population. So this has been done here. This is the evolutionary history. So you have the normal cells here, and they became fewer and fewer, but then you have one clone that expanded and then became smaller, and here is another clone that is expanding and the third clone that is expanding. So it is possible to reconstitute an evolutionary tree like you do when you study species evolution, right? So it's the same approach, the same mathematical models that we are using. And then, of course, the big interest is how does treatment affect this? Because treatment in this paradigm will be equivalent to selection. So we want to try to set up selection pressures through treatment that makes these cancer cells disappear, right? And only the normal cells disappear. But here is another clone of cancer cells, and there is a variant here that unfortunately leads to recurrence of the tumor. So then you would have to hit once more with a new selection pressure that is a new type of treatment. So we feel that it should be possible with these methods to push the cell community back to the normal in the future. And another thing is that we have got now much better methods for precise genome editing, and we are, and most other people are focusing on this type of system, which is the CRISPR-Cas9 system. So this is based on something we have learned from bacteria. It's a defense mechanism in bacteria. So they can recognize a sequence that is not present in their own genome, and when they do that, this RNA molecule is attracting the Cas9 nuclease that binds to this area and cuts in a very precise method so that you destroy this genome target. And this has been developed phenomenally fast. So now we have CRISPR-Cas9 systems that are very specific and efficient, although we know that there are still some off-target effects. So no new treatment is without side effects, so one should be humble still. But this is way more exciting than I myself ever had dreamt of. But it can be used not only to cleave and create genetic changes, and the genetic change can be a knockout, you inactivate this gene, or you can also introduce new genes by homologous recombination. So the genetic changing can be both negative and positive. But now we can also start to make epigenetic changes and thus do precise gene regulation. And this is a new version that we have been working with. You make a Cas9 that is not a nuclease, but it binds in the same way to the guide RNA. And then you can, for instance, like here, you can construct transcription factors that you attach both to the guide RNA, the small guide RNA that we call this part, and to the Cas9 itself. So you end up with a complex like this, that you have all these activating factors, and this means you can activate the gene that lies next to the target sequence that you have used. And you can do the opposite also, so you can block the transcription, so you can repress the expression of specific genes. And moreover, we have started to try to use this to target DNA methylation in the same way. So instead of adding transcription factors, we add a piece of a methylating enzyme. And this is the target sequence with some methylated places and a lot of potential places that are not methylated. And then you add this, so the CRISPR finds this area, and the Cas9 is starting to methylate on both sides. So what you get then is a hypermethylated region, except for the part in which the actual complex is sitting. But to both sides, there is a hypermethylation relative to what you have up here. And this is just to show experimentally that this is the case. These are different constructs, but you can see the blue ones that are functional, they methylate on this side, and they also methylate on the other side. But the potential methylation sites who are here in the middle, they are not affected. And as a control, we can mutate the methylating domain so that it's inactive, and you can see then nothing happens. So it's a specific methylation of potential methylation sites around the target. And this, of course, is very exciting, and although we still have to face off-target methylation, this is so pronounced that I'm sure it can be useful. And then the last part is that not only can we do this with DNA methylation, but we have started to do it with histones also. So we can change the chemical modification of the histone tails in a way that is decided by the targeting of the CRISPR. And this, of course, is very exciting. Although we don't understand much about this yet, we have shown proof of principle anyway. And finally, another group or a couple of other groups have been able to create new loops between this side and this side. So they make a CRISPR that sits here, that is modified so it has a special receptor for a small compound, and another one sitting here that have a receptor for the same compound. When you add the compound, then these two bind to each other, so you create an artificial loop. And although we don't quite know what to do with this, we have seen in these first studies that you can bring an enhancer quite close to the gene and thus activate the gene in the same way as we think is happening in real life. So on top of this, these pigs are, of course, interesting from other points of view. So as you have heard, wireless sensors is being more and more common, so these can be introduced into the pig body to measure different things. For instance, glucose concentration in our diabetic or in our pigs that we study pre-diabetes in. But there are many, many, many other possibilities. And in Aarhus, we also have very, very good facilities for diagnostic imaging with new technologies. Actually, the pigs have better scanners than the patients because these are experimental machines that are developed with the pigs as test materials. So this is non-invasive analysis of early atherosclerosis, and here you have a more invasive method of performing exact diagnostic imaging. So all in all, I think we are in a very exciting time, and I envisage a revolution in medicine, combining the detailed genomic and epigenomic knowledge that we can now obtain very cheaply, and as Yang Huan-Ming said, it's possible to sequence everybody. And then we have advanced biosensors and advanced imaging methods that we can put into work in our bodies. And then we hope to have regenerative medical treatment that can modify and enhance the healthy cells, whether they are from the added or they are cells that are already present in the body. And what is very important, we have to know much better how we select against the damaged cells, because much of the problem of degenerative diseases is that you have poor ability to get rid of all the damaged cells so that you give the healthy cells a chance. And then, not to forget, we should also secure a healthy microbiota, and we hope that our model pigs should help in this process. So with that, I will just show the group that we have done much of this work in Denmark. We call it the Danish Regenerative Engineering Alliance for Medicine, or the Dream Team. And in China, of course, we have the Chinese Regenerative Engineering Alliance for Medicine, so that is the CREAM team. And this is very big, so I can only show you a lot of very, very active and enthusiastic young people who are involved in this. So thank you for your attention.
So now it's a Q&A discussion.
Very, very interesting. Well, for years, we have been using transgenic model, animal model for disease. Now, you introduce to us the epigenetic animal model for diseases. I have three very simple questions. One, procedure-wise, can you compare this? It's much simpler. It's easy to do. That's the first question. Compare the transgenic and epigenetic. Okay. Second, how about pathology and treatment? Make any difference? You talk about off-target. We expect everything, but is there any basic difference? And the third question is, with transgenic, with epigenetics, people say, oh, you can pass on for 3 generations. How about the epigenetic animal model? How many generations can we pass on? 3 questions.
I have to say that we have much more experience with the genetic models than with the epigenetic models, and I can clearly say that this far the genetic models are simpler, and we know that they can be inherited unlimitedly long. So we can create stocks of pigs that are the same for a long period of time. So this means that it's much easier with the epigenetic models. But I still think that the epigenetic models are useful. It will be more a model where you influence the individual pig. I don't think you can hope that the epigenome is inherited to next generations. We know that there are a few epigenetic modifications that are, so there are some imprinting, as you know, and also some externally created variants that are inherited, but in mostly everything is reprogrammed during the production of gametes, and then a new epigenetic program is put on during embryology. So it is a different type of model. And then when it comes to pathology, I think it's too early to say, because we haven't worked very long with the pathology of the epigenetic models. But from our knowledge of diseases related to epigenetics, the pathology is real enough. So although it might not be exactly the same as when we make genetic models, it will still be very interesting.
Hello, I was very interested in your idea of looking for the wellness variants, and you've mentioned about the you were sequencing the Tibetans' DNAs and found the EPAS1 transcription factors. So I was wondering, because, well, according to my limited knowledge, I know that the cancer cells in hypoxia can actually activate the HIF1 alpha protein and triggers angiogenesis. And this is a very distinct feature in cancer cells metastasis. So I was wondering, can we, is it possible that we, by enhancing the, can we activate the patients, like the amount of EPAS1 transcription factor to repress cancer cells induced angiogenesis, and in order to prevent the tumor cells?
Well, it's a good idea, I think, but we haven't tried yet. But it's obvious that if you know a gene that variants can be positive or negative, right? Then I think it can be used in many different contexts. So I think it would be worthwhile, when we understand a little bit more about why the EPAS variant that we have selected for is so good for the Tibetans, that we also can drive it the other way and make it difficult for the cancer cells. But it's a rather remarkable transcription factor variant. As usual, when we do these things, we do not find exactly what we expect, right? So this transcription factor is not only increasing the functions that we need to handle low oxygen, but it actually also reacts in a balanced way, more balanced way. So if it goes too fast and is too acute, the response to anoxia, you get altitude sickness, and this can be dangerous. So not only is this a positive factor, but it's also selected not to overdo it. So it's really a good example of fitness. Fitness is not a selection for the strongest, but to the best adapted, the best. So there is, I think, a long way before we know enough to say that this is a perfect target for fighting cancer. But you're absolutely right that this capacity to increase vascularization and increase oxygen uptake and so on is very important for malignant cell development.
Cancer is much more complicated.
Yes, yes, of course. Yeah. Well, we haven't tried yet, so these are new results, but okay. Yeah.
Hi. Actually, I'm curious about the parts of crystal methane because you show like in different positions they have a different like methylation expression level. So I'm curious about like whether this kind of experiment when you did on different types of cells, is this like the tendency is the similar, or it will have a different methylation expression levels? Thank you.
Well, as I said, methylation is very, very complicated. And lots of methylations seem to be spurious or stochastic and not regulated in a clear fashion during differentiation. So those you can find all over the place, and the heterogeneity is very big. Actually, we are talking about following the clonal development on the basis of epigenetic patterns rather than genetic patterns that we have done right now. But then there are these very important positions that are methylated in a regulated fashion. And these are different from different organs and different cell types. And that's why they have this important effect. So I think they are participating in packing down the DNA that this particular cell does not need for its differentiation, and packs up other parts which is important to be a good cell of this type. So as you know, there are enormous difference between a growing red cell that produces enormous amounts of hemoglobin or a liver cell that produces a lot of detoxifying enzymes and so on. So it's as if they were totally different from each other, but the genetic material is the same. It's the epigenetic material that is different, the epigenetic pattern that is different.
Yes, Lars, both of us are both happy and sad that the audience would be more interested in epigenomics. It means outside of the genome or beyond the genome. Wow, you are even more foresight-seeing.
But of course, we analyze the methylome also by sequencing methods.
On the sequencing.
Sure. You can see where the methyl groups are in genome-wide fashion. So sure. The sequencers will not be obsolete.
We even include epigenomics into the genomics or as a part of genomics. 那位男生，刚才对。
Okay, Professor Bolund, as a medical student, I'm really fascinated by the fact that regenerative medicine can actually come true maybe by HMC method used in the genetically designed pigs. And some of the achievements I've shown here, right, like Alzheimer's, psoriasis, and breast cancer. But I'm really curious about, like, some of these diseases may be really multifactorial. For example, Alzheimer's, there may be some amyloid hypothesis, tau hypothesis, and cholinergic hypothesis, etc. So if the disease is not merely based on genetic issues, how can we deal with that? Yeah.
You're absolutely right that all these diseases that we are working with are multifactorial. So you should just see this as an attempt to make the animals more susceptible, right? So then you choose a gene and a gene variant that you know from the clinic is causing susceptibility, right? So of course what you are studying is atherosclerosis based on this susceptibility gene, but there are also a lot of other effects. So these pigs are also fed very unhealthy diet, right? So and so on. It's true that you cannot get the whole picture, But as we get better, I think we can modify more than one gene. Now with the CRISPR technology, it is possible to, for instance, modify 100 genes. And in this way, you may be able to mimic more the complex genetic background of these chronic diseases, because they clearly are a combination of many gene variants and environmental factors.
谢谢两位 Q&A， 还有呢？
I was hoping class 1 will give you some effect.
Well, we still hope, and we learn something about that. But it's quite interesting because we have been forming a high-altitude consortium with other scientists that have studied similar things later in the Andes mountain and in the Ethiopian mountain. And they also see selection of certain gene variants, but not the same as we see. And so evolution is really complex. So in one place you have a selection for one thing, and in another place you have selection for another. And we just have to learn from all of these examples, right?
哦，这位女生，
So I would like to ask about the CRISPR-Cas9 system, because even if it is improved in the new form, I think there will still be possibilities of unintended mutation for the cells that accept the Cas9, CRISPR-Cas9 intervention. What do you think could be done to improve this side effect of CRISPR technique?
I'm not absolutely sure I understand the question completely, but you are asking about the off-target effects, right? And we know that there are off-target effects, but these are more at a much lower level, right? And also in most cases they have no functional meaning. So I still think that with a very specific and strong effect at the target, you can learn a lot from this. But I give you that you never know quite what is happening. But we can sequence the whole genome and see where are the off-target effects. So in BGI, we have the advantage of really showing what, and we have a paper out, or it's about to be published, which is really carefully analyzing off-target effects, right? So I still think this is very valuable, and as you know, all treatments have side effects, whether they are genetic or pharmaceutical, and gene therapy also suffers from, although you can treat immune deficiencies very efficient with retroviral vectors, there still is the risk that in a few cells the retroviral vector is integrated in a very bad place, so it activates leukemia development, and this is clearly a side effect that one has to take seriously.
Okay, that's the last question, or we will have the last, but not the least question. Okay, please. Okay, 那现在是最后一个了啊。
Hi, Professor. You mentioned in your speech that selection is the driving force in evolution, and maybe treatment can be another type of selection. And I was wondering, can you elaborate more on this idea or give examples that have already be put into practice? Thank you.
Well, usually clinicians do not think in these terms. It's because of my background as a geneticist and an evolutionary biologist that I think in these terms. But I think clinicians are already trying to do exactly this, right? So they are in cancer. They are trying to hit the bad cells and spare the normal cells as much as possible. So this is clearly a selection for the normal cells, right? And when they do not succeed, then they have second-line treatments where they try to hit the recurrent cells still without hitting the normal cells too much. And nowadays, you even have third and fourth line treatments. So you can make people live for a very long time with cancers that should have killed them long time ago. So this is a good thing for the patient, but it's of course a problem for the healthcare system because it costs a lot to do this all the time. But we hope, of course, that the tools will become better. So these cytostatic treatments are not specific and have a lot of unwanted side effects, and the normal cells are not surviving all that well. So the selection is not perfect.
Okay, because of the time, I don't know Lars' talk is better than mine or not as good as mine. You have so many questions because his talk is still not perfect. That's teasing, that's to defend myself. Okay, 那怎么样？那我现在 I will give the floor back to Professor Sung.
Thank you.
Very much.
Yeah. Yeah, thanks very much for Dr. Bolund and also for Professor Yang.